Yeah, I think this is an impactful manuscript. It gives us a really beautiful outline of the CS1 trial, particularly the background for the trial. This is one of the most carefully designed clinical trials that I've participated in because the phenotyping of the patients is so deep. We're using a variety of modalities, including new technology like the CardioMIMS, to really assess the true safety and efficacy of this medication. And this has really panned out well in the manuscript. We go over the history of how we judge medication algorithms. We talk about the contemporary risk scores. We introduce the data about using the CardioMIMS, the first time in a clinical trial where real-time pulmonary pressures are being assessed as we're dosing the patient. This gives us the real first tangible information of when a drug works. We've never seen that with any of the medications that we've used. So sure, we've seen effects on 6-minute walk test and functional class, but remember, those are really late manifestations of a drug's efficacy. What the drug is doing first is it's working on the remodeling of the arteries. And so the pressure changes that we're seeing is really the first thing that happens when a drug works. And we, for the first time, will see when that occurs. And it's interesting, we looked at the data for one patient, and we know when that drug started working. That's fascinating to me, and we don't have that information on any other therapeutic that we use in pulmonary hypertension because we've never assessed daily pressures in these patients. Really important. And the design manuscript really gives us a nice flavor for that. We also go over the unique characteristics of the CS1 compound. This is a very different drug than what we have used in contemporary treatment of pulmonary hypertension. Many of our drugs that we use now have a strong vasodilatory effect and a small remodeling effect on medication. And I think that's the reason why we have to use so many of them in combination to get an effect on the drug. But CS1 is different. It actually has properties that allow the vessel to reshape itself. It works on the essential components of the vessels, not just only the cells, but what's holding the cells together. And it gives us a really good idea how this drug remodels the vessels and tells us some of the other important features of the medication. It has a very strong antithrombotic effect. And this is a unique property that I think might be helpful in pulmonary hypertension because as those vessels restructure in the very small arteriolar space, microthrombi form, and microthrombi are active biological reactors. They release tons of cytokines and growth factors. And because CS1 has this antithrombotic effect, we're hoping that those microthrombi don't form in these patients anymore. And so those little bioreactors are different and really allow the vessel to remodel more importantly.